Welcome to The Explainer. Today, we're diving into something that's happening inside you right now, a biological process that, if it messes up even a little, can have, well, devastating consequences. We're talking about something so tiny, so intricate, yet so critical it can mean the difference between health and disease. Let's get into the wild world of protein folding.
Okay, so what could these possibly have in common? I mean, really, you've got a brain disease, a blood disorder, and then cancer, uncontrolled cell growth. They seem worlds apart, right? Well, the connection between them is a really deep and fascinating biological puzzle.
So the answer, it's hidden in a process that's happening trillions of times every second inside your body. To figure this out, we're going to follow the life of one single super important molecule, the protein. See, to get how these diseases are all connected, you got to understand how proteins are born in the first place. And that story always starts with a blueprint for life, our DNA.
All right, so let's start with the basics. From blueprint to protein in the cellular factory. Seriously, think of your cells as these tiny, incredible factories. They take the master plan, the one stored in your genes, and they just crank out everything. All the enzymes, the antibodies, the bits and pieces that build you and make you work. It's an amazing operation.
This whole process is what scientists call the central dogma of molecular biology. It's basically a three-step flow of information. For what we're talking about today, though, we're going to zoom right in on that final step, translation, because that's where the real action is.
And on this factory floor, you've got a key piece of machinery, the ribosome. The ribosome is the worker on the assembly line. It takes the instructions, that's the messenger RNA, which is like a photocopy of a gene, and it reads it, stringing together amino acids one by one, in the exact right order to build a protein chain.
But here's the thing, and this is so important. What comes off of that assembly line, it isn't a finished protein, not even close. It's just a long, floppy string of amino acids, a polypeptide, if you want to get technical. And in this state, it's completely useless. It can't do a thing.
Okay, so now for the art of the fold, the quality control section. This is the magic moment. How does that useless string transform into a complex three-dimensional machine? Well, that process is called protein folding. And believe me, the cell has a very, very sophisticated quality control system to make sure it goes right.
Just look at the difference here. On the left, you've got that useless string, the brand new polypeptide chain. It's formless, it's inactive. Then look to the right. That is a functional machine. It's been precisely folded into a unique shape that lets it do its specific job. Whether that's carrying oxygen around or fighting off a virus.
So how on earth does a cell make sure every single protein folds correctly and doesn't just end up as a tangled up mess? Seriously, think about it. You've got trillions of these strings being made all the time. The risk of them just bumping into each other and clumping up into a useless or, even worse, a toxic glob is just huge.
And that's where these little heroes come in, molecular chaperones. You can think of them as the cell's quality control inspectors, or maybe personal folding coaches. Their job is to guide the new protein, help it find its correct shape, and prevent disaster. And once the job's done, they're out. They're not part of the final product at all. Okay, so there are different types of these chaperones. First up, you've got HSP70. These guys are basically bodyguards. As that new protein chain is coming off the assembly line, HSP70 jumps in, grabs onto it, and holds it steady. This keeps it from sticking to other proteins it's not supposed to while it's being moved to where it needs to go. Super important.
Then you have this other type, HSP60, or chaperonins. And these are just, wow, look at that structure. It's like a tiny barrel or a double donut. What it does is absolutely incredible. It actually captures a misfolded or unfolded protein inside its central chamber, slams the lid shut, and gives it a private, safe room to try folding again. After about 15 seconds, pop, the lid opens, and hopefully a perfectly folded protein comes out.
So our next section is about when the factories themselves break, what we call ribosomopathies. See, chaperones are awesome for fixing folding mistakes. But what happens if the problem isn't the folding, but the factory that's building the protein in the first place? What happens when the ribosome itself is broken?
This leads us to a whole class of diseases called, you guessed it, ribosomopathies. They're all caused by problems with the ribosome machinery. Now, logically, you'd think, okay, ribosomes are in every cell in the body, so a ribosome disease should mess up everything. But that's not what we see. And that's where it gets really interesting. This table just lays the paradox out so clearly. Take a look. You've got diamond black fan anemia and 5q syndrome. Those mainly hit the blood.
But then Treacher-Collins syndrome affects how the face develops with no blood issues at all. So here's the million-dollar question. If every cell has ribosomes, why do these defects cause such specific, localized problems? It's a huge clue that maybe, just maybe, ribosomes aren't all identical. Maybe they're specialized for different jobs in different parts of the body.
All right, so a broken factory inside a cell is a really, really big deal. It's a major threat. It can lead to all sorts of bad stuff, including cancer. So naturally, the cell has an emergency plan. It's evolved a powerful fail-safe system, a kind of guardian's response.
Okay, so when that ribosome assembly line starts to sputter and jam up because of a defect, the cell absolutely knows something is wrong. It doesn't just carry on. Alarms start blaring internally. The cell senses this danger, and it's called ribosomal stress.
So what happens during this stress? Well, since the ribosomes aren't being built right, you end up with all these leftover parts, free ribosomal proteins just floating around. And these spare parts become signals. They go on a mission. They find and latch on to a specific molecule called MDM2.
Now, why does that matter? Because MDM2 has a day job, and that job is to hunt down and destroy one of the most important proteins in the cell, the tumor suppressor, p53. So when those spare ribosome parts grab onto MDM2, they're basically putting it in handcuffs. MDM2 is neutralized. It can't do its job anymore. And that means p53 is set free. And p53 has a nickname for a very good reason. It's called the guardian of the genome. Once it's activated and off the leash, it doesn't mess around. It hits the big red emergency stop button. It can either freeze the cell cycle, stopping the damaged cell from ever dividing again, or it can go even farther and trigger the cell's self-destruct program. It's an incredibly powerful safety mechanism.
And that leads us to the final and kind of mind-bending piece of this puzzle, the cancer paradox. Hold on. If a broken ribosome activates this super powerful anti-cancer protein, p53, then how on earth can these diseases also increase your risk of getting cancer? It seems like it should do the opposite, right?
This is the paradox, right? From the source. Everybody knows tumors grow like crazy, and to do that, they need to be churning out proteins like there's no tomorrow. They need more ribosomes, more translation. So, how can a disease caused by faulty ribosomes, which actually slows down protein production, somehow lead to cancer? It just doesn't seem to add up.
The answer, it turns out, is all about survival of the sickest. Think about it. Inside a person with one of these diseases, that p53 alarm is going off all the time in their cells. Stop dividing, self-destruct. It's a constant barrage. This creates this intense pressure where almost all the cells are being held in check. But what if one cell, just by random chance, gets another mutation? A mutation that breaks p53? Well, that cell can suddenly ignore all the alarms. It's the only one that can keep growing. The guardian is gone, and now you have a broken factory with no safety inspector, and that is a recipe for cancer. Which just leaves you with this incredible thought, right? How many other diseases that we deal with, how many of them don't start with some big infection or an injury, but with a single invisible submicroscopic error in folding? It's just a stunning reminder that deep inside every one of us, this beautiful, intricate dance of assembly and quality control is happening constantly. And that dance, that's the only thing standing between a healthy life and disaster.